Okay, so as was mentioned, I study energetics, animal energetics, specifically in dairy cattle. For those of you that have taken an undergraduate course in animal nutrition, this figure is probably familiar to you. Basically, what I tell people, whether it's kindergarten classes or world-renowned scientists that come into our lab and I tell them what we're studying, I basically tell them what we do is we measure the energy that goes into the animal and then the energy that goes out and measure where it actually goes. And so we're very interested in studying feed characterization and the chemical composition of feeds, and then of course digestibility. And then as we flow through this chart here, measuring metabolizable energy by accounting for gaseous energy losses, or primarily methane, and then urine, and then understanding the efficiency of utilization of that energy that's remaining, going to pregnancy tissue, milk, or maintenance, and then ultimately getting a representation of net energy for lactation. And over the last 10 years, we've been studying indirect calorimetry, doing really kind of garden variety dairy nutrition studies, but taking these measures of energy utilization and actually retracing some of the assumptions of our traditional feed formulation models. So this is some data that was generated out of our lab. It'll actually be coming out in, no surprise, Journal of Dairy Science shortly. But it's a summary of some data that we've accumulated over the last 10 years. And to me, it really is a nice illustration of where that energy is going after a cow consumes it. And so if you look at the energy in mcals per day on the y-axis, milk, feces, heat, urine, methane, and tissue, and then express it as a percent of gross energy, this is where the energy is going. And so it's no surprise to probably anybody in the room. About a third of that energy is going to the milk. About a third is lost in the feces, and roughly a third is lost in the heat. And then there's smaller portions that are lost in urine, and of course you see methane, which is what we're all interested in hearing about today, and then of course tissue. A couple interesting things to see here is not only the mean of these measures, but you see the variance in our data set. For example, tissue energy, this is just a great illustration just to show you how robust the lactating cow is, how she's able to produce that milk and have such a tremendous flux in tissue energy as she does that. So I want to speak a little more about the methane energy here. Obviously, it represents a small proportion of gross energy, but what we're here today is to talk about the large impact it has on climate change. So this is, again, part of the same data set. I'm not a geneticist, although I'm talking in a genetics section. The reason why, or an easy way for you to figure out why I'm not a geneticist, is I won't talk about heritability. Troy, that was on your first slide, okay? But in this data set, what we did is we broke apart the digestibility of nutrients, and we said, well, of that variance that's observed in the data set, how much of it is attributed to animal? How much of it was attributed to the experiment? How much of it was attributed to the treatment? And then what was residual? And you see these reflected in the error bars here. And so what I have here is the digestibility of NDF, starch, and then dry matter intake. You can see that there's quite a large variance associated with animal on dry matter intake. And that's something that we expected. What we didn't expect is to see this variance due to animal and digestible starch. And you'll notice that variance is actually about twice what it is for digestible NDF. Okay, so what about maybe some of the things that we're interested in here? This is something that really struck us. On that far left-hand side is feed efficiency. And again, I mean, the geneticists have done a tremendous job in helping us breed animals to produce more milk and be more efficient to do that. And I think it's hard for me to see that, but I think it's about 20%. This next one was very surprising to us. This is among animal variants in methane production. So that's the bottom blue chunk of this bar chart. Quite surprising to us, and again, we're not geneticists, so maybe we should have been surprised. This is methane per unit of dry matter intake, okay? So it's not, dry matter intake isn't baked into here. This is methane production per unit of feed consumed. That was almost 40% of the variance in this data set. We'll just go through energy utilization. I know we're less interested in this today, but you'll see the efficiency of use of gross energy for digestible energy, the use of metabolizable energy of DE consumed, and then on the far right, the efficiency of utilization of ME for NEL. And so you can see there is variance around that. It does decrease as you go from left to right, simply because you're accounting for more factors of energy loss as you move through there. So I think one of the reasons why I was invited to this talk was to share just briefly of a research project that we recently got funded from the USDA SAS system. And that's a grant that we've just started collecting data on both beef and dairy cattle. We're running, at least from my perspective, with indirect calorimetry, running large numbers of animals through our indirect calorimeters, where we're measuring obviously methane production, but also measuring digestibility, heat production, milk production, body weight gain, and obviously tissue energy as well. So what we'll be doing is looking at the genetic factors attributed to not only methane production, but the whole cascade of energy utilization. So in addition to those genetic factors, we'll also be looking at the microbiome and trying to identify, again, the genetic factors and the microbial factors that influence whole animal energy and then methane production. And then I always say a good grant has to have a good moonshot. And the moonshot in this grant is to look at early life intervention and actually inoculating younger animals with the microbiome that will result in lower methane production. And that'll be something that we'll explore in the later portion of the grant. So big thanks to USDA NIFA for trusting us with the funds to work on this grant, and we really look forward to moving forward. This is just a picture of the team and the names listed here. I won't go through them there, but they're well represented, all of the fields of genetics, microbiology, extension, and then, of course, nutrition. The second picture there is just a picture of myself and the graduate students at the indirect calorimetry station at Penn State University, the Armsby calorimeter. And so a lot of the measures that we're taking are really old school measures, but we're applying some additional state-of-the-art measures on genomics and microbiology. So thank you very much for the opportunity to share today, and I look forward to the panel discussion.